Good afternoon. This is Carolyn Hiller. I am the program director for the clinical diagnostics program at the MDIC consortium. And this afternoon, I want to welcome you. We have our somatic reference samples, Tumor Suppressor, a webinar that's going on for public comment. The panelists that we have today include JD Alvarez, who's the head of oncology diagnostics at Janssen, and JD is the chair of our somatic reference sample working group. We also have Timothy McDaniel, who is the senior vice president of emerging opportunities at TGen. Tim is on the SRS working group, and he's also a co-chair of our variants working group. We have Michael Morrissey, who's the executive director of oncology genomics and analytics at Merck. He is also on the SRS working group, and he's also our tumor suppressor and gene signature subgroup lead. We also will have Tim Stenzel, who is the director of OIR at CDRH and FDA. And now I'd like to turn it over to JD Alvarez.
Hey, thank you, Carolyn. Yes, so I am JD Alvarez, and I lead oncology diagnostics at Janssen, and I've had the pleasure of chairing the somatic reference samples working group for almost a year now, not quite. So to level set everybody, wanted to introduce you to the MDIC. This was formed in 2013, the first ever public-private partnership whose objective is to advance medical device regulatory science. And so everyone's level set. Regulatory science is the science of developing new tools, standards, or approaches that help assess the safety, efficacy, quality, and performance of FDA-regulated products. This can help medical device manufacturers and government agencies benefit from reduced time and resources necessary for device development and review. But more importantly, patients benefit because it speeds access to important new technologies that may benefit their clinical care. The MDIC's focus is really on regulatory science, not on policy. So we go to the next slide, please. The MDIC has a number of program initiatives which are bucketed into various groups as shown here, all impacting regulatory science. The Somatic Reference Samples Project is one of the projects in the clinical diagnostics program. And you can learn more about this program and other programs by visiting the website, which is shown there in the lower right corner, www.mdic.org. Next slide, please. So the Cancer Genomic Somatic Reference Samples project, the SRS projects for short, is a collaborative effort to develop cancer genomic somatic reference samples. This is specifically for to improve the accuracy, reliability, and transparency of NGS-based oncology tests. The impact, as listed here, is that this will aid in efficient NGS test development and validation. Ideally, this will streamline and possibly remove steps in the regulatory process for diagnostic companies. Do this in a completely transparent way. And most importantly, I think, coming from where I am, this will compress development timelines for targeted therapeutics developers. And again, with the goal that all of this will allow patients faster access to novel therapeutics and novel diagnostics that can enable those therapeutics. Next slide, please. So the SRS project plan is similar to any project that is under the umbrella of MDIC. We brings together a diverse array of stakeholders, includes industry, both diagnostic and the pharma industry in this case, the regulatory agencies, academic medical centers, payers, biorepositories, and professional societies such as the AACR and CAP. Next slide, please. The SRS project has been divided into two phases. The first phase is the phase that we are in right now. This is the scoping of the work. And the goal of this phase is to first publish a detailed description of a prioritized set of samples and variants that will be made available, and to develop a plan for their development and characterization in phase 2. One of the first products that come out of phase 1 is a landscape analysis report, which I'll talk about in a few slides. The landscape analysis working group identified gaps in current somatic reference sample availability. Now, phase 2 of this project will be to pilot, characterize, and scale up. So essentially, we're going to make a plan to contract and oversee the production and characterization of high priority oncology NGS reference samples. And that will be transferred to interested commercial entities. These project outputs will be made widely available to allow broad public accessibility by interested commercial entities. And this is planned to take 1 to 1.5 years. Next slide, please. So in phase 1, when we started out as a large group, we had a number of questions that we needed to focus on. First, we wanted to understand what has been done. There's a lot of efforts ongoing to make reference standards or that are ongoing or have been completed. We needed to determine what are the variants that are most important to develop reference standards for. We had to understand what the characteristics of the samples that will serve as reference standards. And then when we have these, how are we going to validate? How are we going to know that what we've made really are what we think they are? And then finally, how will we make those reference samples available to the broader community? To do this, obviously, there were a number of things we could do, and we decided that in order to make sure that we could get something done, that we needed to focus. So we've started out focusing solely on solid tumors. Hematologic malignancies are currently out of scope, and we focused on tissue standards. Circulating tumor DNA is out of scope for now. So next slide, please. We kicked off last summer, I think it was June, and at that point, we formed 6 working groups, 4 of which started their activities at the outset. There are 2 groups that have completed their project components. First is the landscape analysis, and the second is the variants group, which is maybe a little misspoken. At least part of it's still ongoing. But those 2 groups feed their information into the sample definition working group, which determines what the samples are going to be like. Currently ongoing is sample quality and validation. So it's making a plan for how we're going to guarantee quality and validate the samples that we have. And then additionally, how another working group, the data integration working group, will make a plan of how to take all of the data that we're going to use for validation and integrate it. Finally, there is a business plan working group, which is charged with coming up with a plan to actually create a product or to make a plan so that a commercial entity would be able to make this product. Next slide, please. This is a list of all the various entities that are involved. The core team is myself, Carolyn Hiller is the project director, Giovanna Tezek at CDRH, and Julie Schneider at CEDAR. But we have representation from a number of societies, pharma companies, device manufacturers, government agencies, academic centers, and suppliers, and certainly SpotDX. Next slide, please. What I'd like to do is just introduce you to the landscape analysis, since this is a tangible product that has come out of the effort so far. The landscape analysis work group was led by Barb Zenbauer from Emory. They had a goal of developing a summary to identify other efforts for the development and evaluation of NGS reference samples, which could inform and complement our own goals. Essentially, we need to know what was already ongoing, or what samples existed, so that we weren't going to end up duplicating efforts. So they conducted a comprehensive review of existing NGS reference material products and projects. They surveyed NGS users about their needs, and they put together a summary report of NGS reference materials that is now publicly available. So if we go to the next slide. This is the report. You can download it at that website that is shown there. It's a comprehensive report that has compiled all the information into a format featuring key data fields and URLs, so that you can easily see additional information that you may need. It's been grouped by sample types, such as cells, DNA, RNA, and FFPE. So as I said, the report is available for downloading at the MDIC website. If you can't remember the link, Google SRS Landscape Analysis Report, and it should be the top hit. Next slide, please. So this landscape analysis report has been very useful for us, very impactful. It's allowed us to understand which genes or variants are already available in terms of having reference standards. This was cross-checked against the variant prioritization list, which Tim will talk about in a few minutes. And it tells us which sample types are represented. So by doing this, we were able to determine a prioritization list. For NGS laboratory test developers, this is a nice reference that allows them to quickly see what is available and what is in development, and gives links to sample details and technical contacts. Next slide, please. So the sample definition working group has been chaired by Mary Ellen DeMars at ATCC. They have a number of goals, which is to define, identify the desired somatic reference samples, physical or in silico, but to develop specifications for sample characterization, acceptance criteria, and quality control, and to develop strategies for maintaining the supply and stability of these samples over time. They've made a lot of progress in terms of determining minimal requirements for the reference samples. Things to think about, like consent, availability, licensing, production and distribution. And they're conducting an in-depth evaluation of current samples and attributes from commercial sources and various initiatives that are related to our target areas. So the next slide, please. This is what they've come up so far. For minimal DNA and cell line technical requirements for reference standards, it should be diploid with a normal background. Ideally have a tumor normal pair so that these can be engineered into a tumor sample. These have to be genomically and karyotypically characterized. Enough DNA needs to be produced for a while, which is a very quantitative amount. But essentially, we can't have this. It needs to be abundant enough that that it could be utilized multiple times. Because of that, there have to be plans to minimize the drift over passages. Variant allele fraction was set at least 10%. There should be flexibility to mix this at a custom frequency. And finally, they want to develop strategies to include all the variants that are on our prioritized list, whether that can be done, how will that be done over multiple samples. I think that is my last slide, and I will turn it over to Tim.
All right, thank you, JD. And it's wonderful to be here in front of everybody. I am Tim McDaniel. I'm from TGen, which is an affiliate of City of Hope National Medical Center. And along with Eric Peters at Genentech, I was co-chair of the variants working group for the Somatic Reference Standard Working Group. Next slide, please. So the variants working group, like the wider SRS, has broad representation from a wide swath of stakeholder organizations. And I'm pleased to say that these members have really been engaged, with 1 to 2 dozen individuals regularly attending the meetings and actively participating in producing, debating, and signing off on our work. Next slide. So the group's approach to variant selection was to identify the list of variants or other targets, for example, genomic signatures, that would be covered in an ideal set of reference materials. In order to ensure that this list identified the most useful targets to benefit the most patients today, we drew the targets from the work of objective third parties who didn't have a vested commercial interest in any particular target. So the sources were ones that represent the current standard medical practice, originate from a public sector or independent nonprofit organization, and represent the collective opinion of a broad swath of clinicians and/or laboratory scientists. And what we ultimately settled upon was the following 3 sources: OncoKB, Civic, and the Cancer Genome Interpreter. Next slide. Now, in defining the variants, recognized early on that we were up against 2 fundamentally different tasks, and so we broke the effort into 2 work streams. One of these focused on hotspot mutations, and the other focused on tumor suppressors. For hotspot mutations, the task was simply to define a list of clinically relevant variants, including small variants, copy number variants, and rearrangements, all of which are recurrent in cancer. This effort was led by Dan Klass and Alex Lovejoy at Roche. And as you can imagine, the task of the hotspot team was more straightforward than the tumor suppressor team. And so the team is further along in their efforts. For the tumor suppressor genes, the task is different, as the number of potentially relevant mutations is so large that there would be no hope of representing them all in a reasonable number of reference samples. So the goal would be more to represent a diverse set of mutational classes such that you can use the reference samples to validate that a platform is analytically capable of detecting whatever might come up. The effort is being led by Mike Morrissey at Merck and is the focus of today's seminar. And before handing it over to Mike so he can go into more detail about the tumor suppressor effort, I'll summarize the status of the hotspot effort in the next 2 slides. And next slide, please. With the hotspots, we prioritized our efforts to identify variants with levels 1 and 2 clinical evidence as defined in CDRH's tiering system for NGS targets. Level 1 represents those variants that are associated with approved companion diagnostics, and level 2 represents non-companion diagnostic markers with evidence of clinical significance, such as by inclusion in guidelines or being the target of an agent in registrational trials. And the level 3 variants, which have less evidence of clinical relevance, we generated a list, but really aren't moving them forward for the time being for inclusion in the reference materials. Next slide, please. The process of generating, reviewing, and finalizing the hotspot list occurred over 7 months, with team calls twice a month, 3 face-to-face meetings, a public review period of our draft list, and revision of the list based on this public feedback. The final list was released publicly in January and formed the core of an RFI that was issued to materials suppliers who would ultimately make the reference materials containing these mutations. The RFI closed on April 12th, and we received responses from 5 suppliers, which we're now reviewing to determine invitations for a forthcoming RFA. So that gives you a quick overview of what we did with the hotspot mutations. And with that behind us, I'm going to turn it over to Mike Morrissey to talk about today's main topic.
Thanks very much, Tim. Maybe we can go to the next slide, Carolyn. And actually just go ahead and fill all of this out. I didn't realize that it was animated. So just as a, thank you. So just as a quick review, probably everyone on this call is well aware that tumor suppressors are genes whose protein products protect cells from aberrant growth in a number of contexts cell biologically. Loss of function mutations in tumor suppressors, or loss of function by any mechanism of tumor suppressors, can promote cell growth and advance cells along a carcinogenic path, which typically includes other activating hotspot mutations. And LOF, loss of function mechanisms, include, but are not necessarily limited to, nonsense mutations, indels, especially those that are leading to frame shifts, a limited number of missense mutations, though many missense mutations which are cataloged in tumor suppressors are in fact passenger mutations, gene deletions, and promoter methylation or other mechanisms for suppressing RNA expression. Typically, loss of function must be biallelic. There must be essentially complete loss of protein function in order for a tumor suppressor loss to be tumor promoting, and that essentially allows mixing and matching of the above mechanisms across the patient's two alleles. Next slide, please. And please fill out again. Thank you. So a comprehensive representation, as Tim alluded to a moment ago, of tumor suppressor lesions is essentially impossible. Tumor suppressors are fundamentally different from oncogenes in that most loss of function mutations are non-recurrent. It's easy to insert a premature stop or a frame shift mutation in many different locations along the gene. Although missense loss of function hotspots do exist and should be cataloged, detecting or in this case providing reference samples for only those hotspots would in fact miss most relevant loss of function lesions. So we really are aiming to generate reference samples with a representative set of the types of lesions which would be found in tumor suppressors. Yet we've decided to use clinically relevant tumor suppressors themselves as the scaffold for these tests. Next, please. So our approach to tumor suppressor selection was again similar to the mechanism that was outlined a moment ago, is to select tumor suppressors with clear relevance in clinical practice, for which again a number of representative lesions can be created in reference samples, and the criteria that were used in the tumor suppressor selection working group were a bit different than the hotspot variant group, just because of availability of information. And these included, again, OncoKB, which was used previously, the level 1 FDA-approved companion diagnostics or level 2 standard of care, or used as a selection criterion in ongoing phase 3 clinical trials, so a potential path to a companion diagnostic, or NCCN treatment guidelines. And less stringent levels of evidence, even for instance for tumor suppressors where there's clear biological relevance to cancer, and also tumor suppressors relevant in heme malignancies could be considered in later phases of the project, but we're not considering them now. Next slide, please. So our draft tumor suppressor list fits in one page. This is approximately the number of genes which the SRS collaboration is intending to represent, but doesn't have to be exactly this number. And by using the criteria on the previous page, we came up with this list. So BRCA1, BRCA2, and also ATM, which is the fifth, are relevant in the context of PARP inhibitors, with the BRCA1 and 2 having OncoKB evidence of level 1, essentially companion diagnostic relevance, and ATM under investigation in phase 3 trials. TSC1 and TSC2, again, with the highest level of evidence in OncoKB. And then 2 additional genes with clear relevance in ongoing phase 3 trials, TP53 and P10. TP53 across a number of indications, P10 in breast cancer and potentially other indications. And then 4 genes which are present in NCCN, pardon me for the typo, guidelines. And these are all mismatch repair genes that are associated with essentially causal for MSI indications. And these are a little bit different in that they're not typically genes which are assayed for treatment selection, but are typically instead are typically assayed at germline level to identify patients who need to be routinely and aggressively screened for various types of cancer, including colorectal carcinoma. Next slide, please. Again, I apologize for the animation, which I did not see on my in my version. So we're requesting public comment on bioinformatics strategy for lesion selection. So we're not coming to you today with the specific genomic locations for lesions, but are instead requesting feedback on the identities of the tumor suppressors and the strategy for selecting variants. And right now, our proposed criteria or our proposed process is to generate multiple lesions for tumor suppressor, which should be acceptable in cell lines, given that one loss of function lesion is no less detrimental than 2 or 3 or 4. And we want to represent indels of varying length, so frame shift indels of varying length, up to 25 base pairs, a variety of nonsense mutations, as well as recurrent and likely functional missense mutations that might not be covered by the MDIC variant hotspot effort. And then in order to ensure that at least some of these lesions are present in so-called challenging sequencing contexts, we want to identify some lesions, we want to generate some lesions in GC-rich regions and in regions with proximity to homopolymer runs. So out of scope for phase 1, unfortunately due to complexity, but potentially up for discussion in subsequent phases, will be reference samples which are intended to differentiate between biallelic and monoallelic loss, as well as loss of heterozygosity and promoter methylation and loss of expression at the RNA level. Next slide. Also, it's worth noting that a number of so-called gene signature lesions, so multi gene signatures, were considered and placed out of scope for phase 1, again, due to a variety of reasons, including technical challenges and ongoing efforts by other organizations. We're open to comments on these. So we have excluded microsatellite instability as a direct measurement, even though mismatch repair genes were included among the tumor suppressors listed previously. Tumor mutation burden, there are ongoing standardization efforts driven by Friends of Cancer Research. Homologous recombination deficiency, whether by algorithmic combinations of tumor suppressor loss or LOH patterns across the genome. Base context mutational signatures, such as those published and later refined by Alexandrov and Stratton, and any sort of RNA-based signatures are out of scope at this point. So we really are focusing on what's a list of relevant tumor suppressors and what are the types of lesions that we want to generate in those tumor suppressors in order to be broadly applicable for reference samples representing loss of function mutations more broadly. Next slide, please. So questions for the public comment period. Number 1 are any tumor suppressor genes with required level of clinical evidence missing, and what would be the rationale for inclusion. And sort of a sub-question of this is, what's the appropriate level of evidence for inclusion? So there are a number of genes, including CDKN2A, VHL, RB1, and actually several others, which have perhaps slightly lower levels of clinical evidence in treatment algorithms, but are currently assayed in large-scale basket trials for treatment selection. So should additional genes with this level of evidence be included at this point, if the number of genes doesn't get too large and fits the sample generation criteria. Secondly, we would like comments on the lesion selection criteria. Are there other factors which we would like to make sure that we're capturing besides frame shift indel length, GC-rich regions, and proximity to homopolymer sequences? And lastly, comments on future implementation of multi-gene lesions or signatures as were laid out in the previous slide. Next slide, please. So the process for accessing this list and providing feedback, here's the link. And we request that you review both the list and also the other questions laid out in the previous slide. And please share your comments by May 30th. And next slide, please. And just alternatively, this is available through the MDIC website. It can be accessed there, and you can surf that website and see other aspects of the project which are currently ongoing as well. I believe that's it for my portion. Yes. Thank you.
Thank you. This is Tim Stanzola. It's great to wrap up this webinar before we open it up for questions. It's been a great webinar so far. I want to, state my gratitude for MDIC for leading this entire effort, and for all the stakeholders that have been involved. This is a very important program and project for the community, and the more people that are involved and the more input we get, the stronger the outcome will be, and that's best for everybody. That's good for everybody, and especially for patients. Next slide, please. So just, wanted to reemphasize, my vision, our vision for this, and that is that, we really want to develop a nimble regulatory approach to genomic tests, and this will benefit patients by speeding life-saving technologies to market. And so we want to find ways that really improve the efficiency of the process, and as well as to encourage and speed innovation. So the next slide, the potential uses of these reference samples that are being discussed, and the tumor suppressor list and others, are going to be very useful in developing new tests, especially NGS-based tests. So, and anything that really can help developers, develop their tests and validate their tests is good for us. Is good for the patients and good for this field. This is really meant to supplement clinical samples that have been largely the mainstay of validation, both analytical and obviously clinical, and it is a baseline. But it really, these will in particular supplement those variants that are hard to find, and provide ready access to these kind of samples to basically the whole community. So, so making sure that we find the right variants to include in this collection is very important. And then characterizing them well is also very important. We'd love for this this collection to be transparent, and for the approaches to be harmonized. And of course, there's many other utilities for this. Already the landscape analysis that's been completed has been a great advantage to the field, we believe, as well. Next slide. So to summarize what we've covered today, MDIC is a public-private partnership. We see this as a great way to engage all the stakeholders, and we're really pleased to be a part of it. There was an overview of the Somatic reference sample project, and then, specifically today, focusing on tumor suppressor genes and the variants of importance, was reviewed what the process and criteria are for prioritizing those variants, and the genes to be considered for reference samples. We reviewed the prioritized list of level 1 and level 2 tumor suppressors. We'd love for the community to take a look at what's been done and to comment. So this is where really engaging as many stakeholders as possible would be awesome. Next slide. So again, you can access the gene list here. You can submit your comments to the email address in bullet 2. And please reference the gene name for any comments on existing genes. And this period again is open until May 30th. And with that, thanks so much, and we'll open it up for questions. Next slide.
Thank you very much to the panelists. And if anybody online has any questions, you're welcome to submit them in the chat box. Otherwise, please email your responses and any questions that you have to the email inbox of srs-tsgs@mdic.org. Okay, I don't see any questions coming up in the chat box. Panelists, is there any statements or topics that you would like to make sure that folks recognize?
Hey, Carolyn, this is Tim. Oh, go ahead.
No, I was just going to say, I think I just wanted to emphasize that we really appreciate the feedback that we can get broadly across the scientific community, and so please submit comments and also spread the word if you believe that others would have useful comments as well.
Thanks, Mike. Tim, do you have any closing statements?
Nothing to add to what Mike just said. Thank you.
Okay. Tim, JD, anything that you want to add before we wrap it up?
Nothing, just echoing what Mike said. You know, we really need public comment from this.
Same here. Thank you.
Okay. Thank you very much. This webinar has been recorded and will be available on the website that you see listed here, MDIC.org, Project Cancer Genomic Somatic Reference Samples. Thank you very much, and I wish everybody a wonderful day.
Thank you.
Thanks, Carolyn. Thanks, everyone.